Hi, my name is Tusita. Welcome to my biology class. If you need more support, log into my website www.tulipdust.com. Also, you can reach me via my telephone number. Today, I am going to talk about the protein synthesis. Protein synthesis is a very important process of all the cells. You can remember that we have learned nucleus has two functions. That means it controls all the activities of the cells. Second thing is the inheritance. That means passing genetic information from one generation to the next generation. Parents to the offspring. And then next generation. When we talk about the control, how does it control? It stays in the nucleus. It never goes out of the cells, even for holiday. So now control is done through proteins. That means official representative of the DNA to do all the function of the cells. The assigned person is the proteins. Let's say this is a lysosome and it contains the red color is the protein, that is enzyme. When lysosome start phagocytosis, that's a phagosome as a bacteria, and then enzyme is secreted into here and they digest. That's a phagocytic cells. Let's say beta cells of the pancreas, what does it do? It just secretes exocytosis, the insulin. And insulin from distal part of the cells, that is not in the cells but maybe other side of the cells, it does its function. That is controlling the glucose level. So how does the process take place? The protein synthesis process takes place in two steps. The first stage is transcription. Second one is translation. In my previous video, I have shown you what is transcription. Today, I am going to talk about the translation process. So before start the translation process, simply I summarize what is transcription. During the transcription, inside the nucleus, the DNA is unwind and then open up the relevant gene which should be transcribed or expressed. So let's say this part, the yellow part, represents the DNA. This DNA is represent the gene. Gene represents a particular protein. So this one is copied into the mRNA by transcription. That's called antisense strand. That is the sequence copied to mRNA. The strand is template for mRNA. After that, this mRNA is leaving out of the nucleus through nuclear pores. As a tiny pores could be around 100 nanometer. There are windows, just molecules can come and go out. So now this one, the molecule is from here, inside the nucleus, it's moving out to the cytoplasm. So let's see this video, that process very quickly. That's a nucleus, and that's a nuclear pore. You can see the messenger RNA molecules coming out of the nuclear pore into the cytoplasm. So now they are moving to the cytoplasm. So now today our lesson is to discuss in detail translation process. During the translation, the first step is that's our mRNA, newly arrived from the nucleus, waiting to make the proteins. The first step is the assembly of ribosome and the tRNA molecules. Showing you the tRNA molecule. So this tRNA molecule is already activated by attaching the amino acid to here. This is called anticodon region. And this is a codon. Codon is triplet of nucleotide. A codon is representing one amino acid. Now first thing, the small subunit of ribosome assembled to the region. This region is start codon. Why is that? The first one is AUG. AUG make methionine as well as it's a start codon. So this tRNA molecule now binds to the codon here. That's a complementary base pairing and hydrogen bonds are made. If it is AUG on mRNA, the complementary base pairing is A is U, UA, this is C. The tRNA molecule has UAC and they make the hydrogen bond like this. The whole process is called initiation. That's the number one process, first stage. First stage is initiation. Small subunit bind of the ribosome bind to the start codon region. At the same time, tRNA molecule and come and bind to the start codon AUG. So we're going to see that one again here in this video. Okay, now you can see that's a mRNA molecule. This is small subunit of the ribosome. Here is a codon, AUG, start codon. Now this one is a tRNA, it brings methionine. That's a complementary base pairing and making the hydrogen bond. And then the large subunit come and bind. Let's complete the initiation stage. So let's complete the initiation stage. The large subunit of ribosome bind here. So, summary of the initiation stage. Small subunit bind, tRNA bind to the initiation codon, start codon. Let's move on to the second stage. Second stage is elongation. This is the mRNA and this one is the ribosome, large and small subunit, and here the tRNA is already attached to the mRNA. So this has 1, 2, 3. This has 1, 2, 3. 1, 2, 3. That means inside the ribosome we can see 3 codons here, here, here. And these 3 regions are named as E region, P and A. Next, another tRNA which comes and again bind to this region by complementary base pairing. They make the hydrogen bond here. Then these two amino acids make the bond. The bond is peptide bond. And the reaction is called condensation reaction. Next, the whole ribosome is moving just one codon this direction. That means I'm going to show this one in this red color after it moves. That means the first tRNA move to the E site and it exits. And this one now in the P site and A site is vacant. Next, to the A site, new tRNA can come and bind. And they're going to join again by condensation reaction to attach the third amino acid. So this process is continued until it completes all the codons binding with the tRNA. This is called elongation. So let's see again elongation here. That's an A site, that's a P site, that's an E site. Now P site is already occupied by the start codon, that is the tRNA with methionine. Now A site, new tRNA is arriving. It brings new amino acid. And then it makes the complementary base pairing, the hydrogen bonds are made. And then methionine and this new amino acid, they bind together by condensation reaction. This is called peptide bond. Now it's moving one nucleotide. E site exit, P site vacant. The P site is already this new tRNA is here, and A site new tRNA join, and they're going to make a new peptide bond. So this process continues until all the codons are ready. So new one is coming to the A site, bind, and they make a new amino acid attached to here, is leaving. So this process continues until the third stage comes. The third stage is termination. During termination, if the tRNA find the stop codons, so these are the stop codons. When the tRNA molecule bind one of these stop codons, they're going to terminate the process and the subunits are disassembled, and then this polypeptide chain is released. So we're going to check this one here. That's still elongation. So now termination start. And then the termination point tRNA bind, the whole complex is released. The polypeptide chain release after that. This is disassembled. So translation has initiation, elongation, termination. During initiation, small subunit and the tRNA start codon attached to make the complex. Then the large subunit bind. Elongation, new tRNA brings and make the condensation reaction to make the polypeptide chain. Termination, the termination codon reach, the whole process disassembled and all molecules are separated, polypeptide chain is formed. Okay, thank you very much for joining my class. So let's meet again in another biology class.